Right. Good afternoon, everybody. Thank you so much for joining us for today's Neuroimaging Summer Series, where we showcase the cutting edge research of the neuroimaging faculty and affiliates at the Michigan Alzheimer's Disease Research Center. My name is Stephanie, and I will be monitoring things here on the back end. If you have any questions or comments, feel free to put them in either the Q&A or the chat. Towards the end of this talk, I will be sending out a Qualtrics evaluation, so please take the time to fill out the evaluations. We really value what you have to say, any topics that you will be interested in learning about in the future. And also, mark your calendars. Our next neuroimaging summer series will be August 7th at 3 p.m. Dr. Shruti Mishra will be presenting Probing White Matter Microstructure with Diffusion-Weighted MRI Techniques and Applications in ADRD. So we hope you can register, and we'll see you all there. And without further ado, it's my privilege to introduce Dr. Patrick Pruitt. He received his PhD in neuroscience at the University of Michigan in 2016. He completed postdoctoral fellowships with Yeski Damiso at Wayne State University and Hiroko Judge at Oregon Health and Science University. Following a position at the Alzheimer's Clinical and Translational Research Unit at Massachusetts General Hospital, he returned to the University of Michigan and is currently the Imaging Research Specialist at the Research Program on Cognition and Neuromodulation-Based Interventions. And with that, I will turn things over to Dr. Pruitt.
Thank you so much. Thank you for the invitation to speak here today. I'm really thrilled for the opportunity. And before we begin, I just want to note that the word primer was chosen very intentionally in the title of the talk. So this talk is deliberately aimed at an audience who may be interested or curious to learn more about brain connectivity, but doesn't necessarily have a neuroimaging background or neuroimaging expertise. So that could be clinicians or researchers who have expertise in other aspects of cognitive aging and dementia and are potentially interested in incorporating neuroimaging data into their work. But that could also be, I know on the webinar, we have students and RAs and techs who work really hard to help our labs and research programs collect neuroimaging data. But for very understandable reasons, they don't always get the opportunity to learn about how these data are used and how they can help address our research questions. So the talk is really designed a bit more with them in mind. There we go. Okay, so with that in mind, we're gonna start with a quick refresher on the basics and work our way forward, starting with the neuron. So our brains contain billions of specialized cells called neurons that can send and receive information. So here we have the cell body. The cell body, where the nucleus of the cell is located. And these branch-like protrusions around the cell body are the dendrites, which act like antenna for the neuron. So they receive signals from other neurons and then pass those signals along to the cell body. The neuron also has a long, slender protrusion from the cell body called the axon, which can connect to the dendrites of the other neurons to pass along signals. And the place where the axon terminal of one neuron meets the dendrite of another neuron is called a synapse. So when a neuron fires an action potential, an electrical pulse travels down the axon to the axon terminal, where neurotransmitters are released into the synapse. So we'll zoom in on a synapse to see what that looks like. So again, thinking that an electrical pulse has traveled down the axon here to the axon terminal on the left, and this leads to the release of neurotransmitters into the synapse. Some of these neurotransmitters diffuse across the synaptic cleft and bind to the receptors of the postsynaptic cell. And depending on the receptor, this may make the postsynaptic neuron more or less likely to fire an action potential itself. Thus, one neuron has passed a signal onto another neuron, and we're looking at really the most fundamental unit of brain connectivity here. But of course, in an MRI scanner, we're not able to measure connectivity at this level of granularity. So returning back to our presynaptic neuron, note that the axon is wrapped in a fatty material called myelin, which covers segments of the axon with small gaps in between. So why is the axon wrapped in myelin? Well, it turns out that the presence of myelin really increases the speed at which the electrical signal can pass down the axon, meaning that the neuron can signal other neurons more rapidly than it could without the myelin. So you can think of the axon as sort of like an electrical wire with the myelin providing insulation. So just to reiterate, the neuron signals other neurons in part by sending an electrical signal down the axon, and the speed of that signal is greatly enhanced by myelin. Now, zooming out and looking at gross brain anatomy, here we have a coronal slice of brain. And after a quick visual inspection, you can notice that a few things stand out. One is that there's a part that's darker in color and goes around the outside edge of the brain, sort of like bark around a tree trunk. And then more internal to that, there's another part that's lighter in color beneath the surface. And finally, as we go even further internally, there's a set of structures that are again colored darker. So the outer part of the brain is the cerebral cortex or cortical ribbon. As you can see, it's like a ribbon that winds its way around the outside of the brain. These structures on the inside are subcortical structures, and these include regions like the hippocampus, the thalamus, the amygdala, and so on. And collectively, we refer to these darker parts of the brain, the cortex and the subcortical regions, as gray matter. And so the gray matter is primarily composed of the cell bodies of neurons with relatively fewer axons. And the other lighter portion is the white matter, which is primarily composed of axons with relatively few cell bodies. So remember that the axons are sheathed in the fatty white myelin, which gives the white matter in the brain its characteristic lighter color. And before we move on, I'll just also note the ventricles, these cavities in the brain that store cerebrospinal fluid, because they'll be mentioned briefly later. So the cerebrospinal fluid surrounds your brain and provides a cushion to protect it from trauma. Now, going back to looking at the gray matter and the white matter, importantly, if you have neurons that are projecting their axons to the same remote location, those axons can group together in bundles that we refer to as white matter tracts. And these tracts allow effective communication between spatially remote brain regions. So for example, in this illustration, we can see the superior longitudinal fasciculus, this large bundle with lots of axons, connecting the cortex in the front or anterior portion of the brain with the cortex in the back or posterior of the brain. So now, with the anatomy in place, how does this relate to brain function? So over time, our field has made great progress in understanding how the brain is functionally specialized. That is, how different brain regions are responsible for different aspects of behavior and cognition. I understand the text here is a little bit small, but really I just use this figure as a stand-in for the type of image that we might see in like the brain and behavior lecture of our intro psych course, or the chapter of our intro psych textbook, where it talks about how the frontal lobe does this and the parietal lobe does this, and so on. And so a lot of this was originally figured out with lesion studies, where a part of the brain was damaged or removed, and doctors would observe how that changed the behavior of the patient. Sort of a classic example, when patient HM had a large section of his bilateral medial temporal lobes removed in order to cure his epilepsy, and he develops anterograde amnesia, This then suggests that the medial temporal lobes, and in particular the hippocampi, play a critical role in the formation of new episodic memories. And further progress was made in our understanding of this functional specialization with the development of various forms of neuroimaging, where participants would engage in a particular cognitive task in the scanner, and then researchers could observe which regions of the brain show increased response during that task or in response to particular stimuli. So now we have the fusiform face area, which seems to respond preferentially to facies, and the parahippocampal place area, which seems to respond preferentially to places. So we are now able to further identify regions that seem specialized for particular aspects of cognition. But we also know, if for no other reason than just looking at the anatomy that we just reviewed, that brain regions are deeply interconnected. So while they each may be involved in particular behaviors, we also know that they're not solely responsible for those behaviors. And so behavior and cognition rely on the effective communication between different parts of the brain. And so in contrast to functional specialization, where we focus on individual regions and the parts that they play, tying them to specific aspects of cognition, here we have functional integration, looking at how brain regions connect with one another and how those connections and networks underlie behavior and cognition. And this is really a critical shift in emphasis. So taking our knowledge of functional specialization and building on it in order to examine how brain regions are connected with one another and form a more complex model and better understand how the brain underlies behavior. And it turns out that that shift in emphasis has been a real leap forward for cognitive and clinical neuroscience. So with that said, let's dig into this idea of brain connectivity a bit more. So what are some of the ways that we can characterize brain connectivity, and how do they differ from? Let's think about two cities. So for our example, we'll use Ann Arbor and Detroit. And let's say that you want to think about how these two cities are connected. So let's say maybe you're in Ann Arbor. You've been hearing about lots of exciting things taking place in Detroit. You heard that they just reopened that beautiful train station. It looks great. The NFL draft was a big success. A few months ago, I actually did a kayaking tour on the Detroit River under the full moon. It was exquisite. Would highly recommend it. So let's say you heard about this fantastic kayak tour. You're in Ann Arbor. You're hearing about all these cool things in Detroit, and you want to know more about how the two cities are connected. Well, connected could mean a few different things, right? So if you're a very literal person, one basic question you might ask is there a road that goes between these two cities, right? So answering that would give you some sense of how the two cities are connected. Another question you might be interested in is, what kind of shape is that road in? If I want to know about the connection of the two cities, if I'm thinking about traveling between them, I want to know, are the roads between them in good shape? Are there problem areas I need to look out for, potholes, construction? That interchange between 275 and M14 has been the bane of my existence for the past month or so. Going to be really glad when that gets cleared up. But so that helps us get a sense of a structural connection between these two cities. So note these two questions: is there a road between the two places, and what kind of shape is the road in, are asking purely about the structural connection, just the physical infrastructure that links these two places. And these are the exact types of questions that are addressed when we investigate structural connectivity in the brain. If we have two brain regions of interest, for example, we can ask, what is the white matter pathway that connects these two regions? And what kind of shape is that white matter in? Or put another way, what is the integrity of the white matter that connects them? And we can answer these questions using an MRI sequence called diffusion-weighted imaging. And the reason it's called that is because it leverages the diffusion of water molecules in the brain to tell us about brain structure. And it's especially useful for informing us about white matter pathways. So how does the diffusion of water molecules tell us about white matter pathways in the brain? Well, remember when we were looking at that slice of brain and I mentioned the ventricles that contain the cerebrospinal fluid? Well, think about water molecules in that ventricular space surrounded by cerebrospinal fluid. Those water molecules have nothing restricting their movement, so they are going to be equally likely to diffuse in any given direction. Now, instead, think about water molecules in a white matter tract, so composed of a bundle of axons moving in a specific direction. And remember that these axons are wrapped in a fatty myelin sheath. So unlike in the cerebrospinal fluid, the water molecules in the white matter are more restricted in the direction that they will preferentially diffuse. They're going to primarily diffuse along the axis or the direction of the axons and are going to be less likely to diffuse perpendicular to the axis of the axons. So when we observe highly directional diffusion of water molecules, we can then infer the presence and direction of white matter pathways in the brain. And this is the principle that underlies diffusion-weighted imaging. So going back to our questions about structural connectivity, one was, is there a path that goes between these two places? And we can answer that question with diffusion data using an approach called tractography. So tractography uses the diffusion data to estimate the trajectories of various white matter tracts, effectively mapping out the major white matter pathways in the brain and allowing us to see what is connected to what via white matter. Now, this image is oriented in the sagittal plane, so you're essentially like looking at the side of the head or the side of the brain. And it's the colors, it's color coded according to an atlas, such that the strands are assigned to known white matter pathways in the brain. And this is another example showing white matter pathways characterized by diffusion-weighted tractography shown from a different angle, in this case, a coronal view. And in this image, the fibers are color-coded based on the primary direction in which they are oriented. So you can see the reddish fibers here in the middle are primarily oriented between right and left, basically going in the direction from one ear to the other, one hemisphere to the other. The green fibers are primarily oriented between anterior and posterior, basically going between the front and back of the brain. And then the blue fibers are more primarily oriented between superior and inferior, so the direction going between the top and bottom of your brain. So having mapped out the white matter pathways of the brain using tractography, you can then look for fibers that go between and connect your two regions of interest. The second question that we asked about structural connectivity is, in how good a shape is the pathway between these two places? And we can begin to get a sense of the answer to this question with another diffusion-weighted imaging derived metric called fractional anisotropy. So remember we saw earlier how water molecules in cerebrospinal fluid are free to diffuse equally in all directions. And this property of being equal in all directions is called isotropic. We also saw that in white matter pathways, the diffusion of water molecules is restricted. Their propensity to diffuse is not equal in all directions and is therefore referred to as anisotropic. So rather than being equal in all directions, it's more directional. And part of what keeps water molecules diffusing along the direction of the axon is that myelin sheath that goes around the axon. So the myelin helps to keep diffusion here anisotropic. But imagine that the myelin along a given white matter pathway was damaged, and it's not serving as an effective insulator for its axon. So we can consider two potential consequences of that. One is that damage to the myelin means that the electrical signals that are propagating down that axon are not going to be able to move as quickly. Because remember, it's that intact myelin that helps to speed up the electrical messaging. So damaged myelin might likely means slower signaling. Another consequence is that the water molecules here will be somewhat less restricted in their diffusion. So they'll be more likely to diffuse against the direction of the axon, making their diffusion a little more isotropic and a little less anisotropic. And this is the assumption that fractional anisotropy works off. So you can use diffusion-weighted imaging to calculate a map of the white matter where each voxel has a measurement of the anisotropy of diffusion. So in this fractional anisotropy map that I'm showing now, the brighter spots represent the areas with the greatest anisotropy. So again, in the places where anisotropy is high, we can infer that the myelin is more intact, or the white matter microstructure is in better shape. In the places that are dimmer, that have lower anisotropy, we could infer that the integrity of the white matter in those locations is reduced. Now, whether those are correct assumptions and appropriate inferences is a matter of some debate, but regardless, fractional anisotropy is a commonly used metric to characterize white matter integrity. So we can collect maps like this across participants and then use them to compare fractional anisotropy throughout the brain across different groups or across different ages, or see how it changes over time. So again, it's a commonly used measurement that we use to infer white matter integrity. So our two measurements of structural connectivity, tractography gives us a map of the location and direction of the white matter pathways in the brain, while fractional anisotropy gives us a measurement of the integrity of the white matter across the brain. Okay, so now let's go back to our example of two cities, Ann Arbor and Detroit, and the connection between them. So this time, maybe we're interested in a different type of connection. So not just the infrastructure that's physically between them, but more in how the behavior of the people in the two cities are connected. How similarly do the people of Ann Arbor and the people of Detroit behave over time? So this is a bit arguably more of a subtle and complex question, a subtle and complex type of connectivity. It's not structural in nature. And when we ask this type of question in the context of brain regions, we refer to it as functional connectivity. So when we ask how two brain regions are functionally connected, we're asking how similarly do they behave over time? And then we use that similarity as a proxy, or we assume it gives us some information about how these two brain regions communicate and how strongly they're in communication with one another. So this type of question can't be answered by looking at structural measures. So just like questions about the similarity between the people of Ann Arbor and Detroit, that question can't be answered by looking at the asphalt of I-96 and M14. We need a different approach to measure functional connectivity. And the most widespread approach is functional magnetic resonance imaging, or fMRI. So fMRI relies on the BOLD signal, which is short for blood oxygen level dependent signal. So when neurons fire, they need delivery of oxygen to replenish their energy stores and be able to continue functioning. So blood delivers oxygen at a greater rate to an active brain region compared to a less active brain region. because the magnetic properties of oxygenated and deoxygenated blood are different, we can use a magnetic field, like the one in an MRI machine, to measure where oxygenated blood is being delivered. And from that, we can infer that those regions are active. So fMRI tracks this signal over time, so we can see how activity changes across the brain or in a particular brain region, and then use this measurement to see how a brain region is functionally connected to other parts of the brain. So if we're interested in calculating functional connectivity, we may start with a particular region of interest and that we referred to as a seed region. And we extract a time series of the bold signal. So essentially, the measurement of the bold signal, that we use as a proxy for activity over time. And if we're interested in learning how a brain region is functionally connected with the rest of the brain, what we can do is then look at all the other voxels in the brain and extract similar bold time courses from those voxels, or it could also be a specific region of interest. And we look at the correlation between those two time series. So this could be positive or it could be negative. So where the correlation is positive, that means that when the bold signal goes up in one region, it also goes up in the other region. When it goes down in one region, it goes down in the other region. So the bold response in these two regions is positively correlated. There are also cases where you might see a negative correlation between the bold time series of different regions. So when bold response goes up in one region, it consistently goes down in the other region. And when it goes down in the first region, it goes up in the other region. So when might this be the case? When might we see a negative correlation between bold time courses? So one example could be if there's top-down modulation, by prefrontal cortex of a subcortical structure, such that when activity in the prefrontal cortex goes up, it leads to an inhibited response in the subcortical structure. That would be observed as a negative correlation in their bold responses. So we calculate the correlation coefficient between the time series between our seed region and every other voxel in the brain. So whether it's positively correlated, negatively correlated, there's no relationship at all. We get that measurement for every voxel in the brain. We transform it to a z-score, so we have a statistical value for each voxel. And then we can apply those values, put them in a statistical map. So you can very easily see the regions where there is meaningful positive connectivity or positive correlation between the time course of your seed region and the colored blobs, or negative connectivity, which would be in the bluer colors. And so these statistical maps provide a very simple visual aid to see where in the brain there is functional connectivity between your seed region and the rest of the brain. And really, at its heart, this is the core of functional connectivity, this correlation between time series of different regions in the brain. Building off of this principle, in 2006, Jessica Damaso and colleagues published some astounding results. Using functional connectivity, they were able to identify several distinct brain regions. So brain networks, distinct brain networks. And so these are groups of brain regions that were reliably connected in each participant. And now, what is it, 18 years later, we take the network organization of the brain somewhat for granted, but the identification and reliable replication of these brain networks really helped mark a turning point, or push the field forward in how we think about brain function, how we think about brain connectivity and how the brain is organized, and how that supports behavior and cognition. We're no longer thinking only about singular brain regions, but networks of regions and their connections. So in the figure on the slide here, you may have heard of some of these networks, like the default mode network, executive control network, salience network, and dorsal attention network, which are generally linked to different aspects of behavior and cognition. So now we have these two types of brain connectivity: structural connectivity measured with diffusion-weighted imaging, and functional connectivity measured with functional MRI, which led to the identification of functional brain networks. And now the relevant question is, what can these measures of brain connectivity tell us about aging and neurodegenerative disease? So starting with diffusion-weighted imaging, unsurprisingly, comparisons of diffusion data between younger and older adults show reduced white matter integrity in older adults, suggesting a breakdown of the white matter microstructure as we age. Excuse me. In the 2007 paper by Grieve and colleagues, this reduction was especially pronounced in the prefrontal cortex. So clusters on the map show where the areas where fractional anisotropy was more negatively correlated with age. That is, areas of the greatest age-related reductions in white matter integrity. And we also see that in this top graph, showing the association between age and fractional anisotropy. Furthermore, the degree of reduction in white matter integrity was associated with reductions in cognitive performance. And that's what the lower graph is showing. So these data are correlational, but what they suggest is the possibility that age-related breakdown in white matter microstructure, an effective reduction in brain connectivity, may play a role in reduced cognitive performance in aging. Now, turning to functional connectivity, if we think back to the network organization of the brain, I mentioned several brain networks that are found reliably between individuals. And if we think about looking at functional connectivity from the perspective of network organization, there are a few different things we may be interested in. One is the strength of functional connectivity within regions of the same network. So we can see, if we say that the different circles in red are regions of one network, the circles in blue are regions of another network, we may be interested in how the blue regions connect with other blue regions and how the red regions connect with other red regions. Those would be measures of within network connectivity. But we can also be interested in measures of between network connectivity, so the functional connectivity between regions of different networks. And it turns out that in younger adults, within network functional connectivity is relatively high, while between network connectivity is relatively low. And another way of saying this is that the networks are highly segregated. And researchers use this ratio of within and between network connectivity in order to calculate functional segregation of brain networks. And it's thought that this network segregation helps to support healthy cognition by allowing networks to specialize, They're strongly connected within themselves and more weakly connected across networks. But as we age, within network functional connectivity is relatively reduced, while between network connectivity increases. So this marks a loss of network segregation that could reflect reduced specialization of network function in older adults, which may contribute to impaired cognitive performance. Beyond normal aging, functional connectivity may also be an early indicator of future cognitive impairment or dementia. So using data from the Alzheimer's Disease Neuroimaging Initiative, or ADNI, Jones and colleagues identified what they characterized as a series of connectivity overloads, starting with the failure of the posterior default mode network, and then cascading through the brain in a series of brain network failures that accompany cognitive decline and dementia. And interestingly, this posterior default mode network failure preceded measurable accumulation of amyloid, suggesting that it occurs early in what we currently conceptualize as the progression of Alzheimer's disease. So if we look at the Jack curves here, so along the x-axis you have the progression of clinical disease from cognitively normal to mild cognitive impairment to dementia. And then along the y-axis, you have the degree of different biomarkers. So starting with amyloid beta and tau, and then changes in brain structure, and then later on, the memory impairments appear and the reductions in clinical function. And again, what Jones and colleagues demonstrated is that these failures in functional connectivity, these network failures that sort of start the cascade, seem to precede even the measurable accumulation of amyloid beta, suggesting that these changes in functional connectivity occur relatively early in disease progression, making them potentially very valuable to investigate if we're interested in trying to predict individuals who may be susceptible to future cognitive decline. So now I want to give an example of incorporating brain connectivity into research in order to deepen understanding of cognitive aging and impairment. So this is a project that I led with mentorship by Hiroko Dodge and Jessica Damozo, and was supported by the MADRC. So this work was funded through one of their pilot grants and used MADRC data. It was recently published in Journal of Alzheimer's Disease Reports. And the origins of this project started with Hiroko. So she's done some really great work in the past, looking at social engagement as a protective factor against cognitive decline in older adults. So that's really the core concept that this project started with. So older adults are particularly vulnerable to social isolation, which can also be phrased as a lack of social engagement. There are a variety of adverse health effects that are associated with social isolation, physical, emotional, and psychiatric, and cognitive health effects. And this can manifest in a variety of different ways. Lack of tangible support, access to medical care, for example, as well as a lack of intangible support, like emotional support or cognitive stimulation. On the other hand, those older adults who have larger social networks, higher social engagement, that seems to confer, or at least be associated with, protection against dementia risk. And two individuals, given the same level of disease burden of AD pathology, those with a larger social network have a better cognitive trajectory. So potentially some sort of increased reserve against pathological insult. And so Hiroko's work, among others, had demonstrated a positive association between social engagement and cognitive performance. So to start with, we wanted to begin by seeing if we could replicate that effect. And so in order to do that, our measure of social engagement, we use the Lubin Social Network Scale, 6 items. These are 6 questions where participants quantify the size of their social networks. So they're asked questions about, friends and family members. How many individuals do you see at a certain frequency? As well as other questions that sort of get more at the relationship between themselves and their social contacts. Uh, how many people do you have that you would trust talking to about a particular issue? Um, and for our measure of cognitive performance, we use the UDS-3 Global Composite Score. So this is a composite of several different aspects of cognition, executive function, memory, attention, and so on. And so we worked with MADRC and received data for 112 older adults without dementia around age 70. And their Lubin score total, their Lubin total score, the total of the 6 items, was just shy of 20. And to give you a reference point, I believe the cutoff for social isolation is 12. So these were relatively socially engaged individuals on average. So again, our initial question of interest is replicating this effect between social engagement and cognitive performance. And it, didn't blow the house down, but we did find a small but statistically significant association between the total, the Lubin Social Network Scale, and our UDS-3 Global Composite Score, which led us to our next question. And this was really, where Hiroko had come to us and was interested in, using neuroimaging to deepen or arrive at a more complete answer to this question. And that was, how does brain function relate to social engagement and cognitive performance in older adults? So beyond just the relationship between these 2 measures, is there a brain measurement? Is there some measure of functional connectivity that helps us to better understand this relationship? And so we started by looking at relationships between individual differences in social engagement and the Lubin score, and within network functional connectivity of 2 brain networks. So we focused on the default mode network and the salience network. We hypothesized that these were the networks that would be most sensitive to social engagement. And again, we found a small but statistically significant association between social engagement, a positive association, so increased social engagement and increased within network salience network functional connectivity. And so then we began to move forward to the next question, and we're interested in looking at a mediation model, whether salience network functional connectivity potentially mediated this relationship between social engagement and cognitive performance. And I won't keep you all in suspense. In fact, it did. Salience network functional connectivity was a mediator of the relationship between social engagement and cognitive function, and mediated 31.9% of the total effect, which is, is not nothing. It really speaks to the role of the salience network in behavior and cognition and in social behavior. There are two additional notes that I do want to make here. One was that at a reviewer's request, we split up the Lubin scale into questions about contact frequency and questions on what, for lack of a better term, we referred to as contact intimacy. So again, there were some questions in the scale that were more about, how often do you see someone? How many people do you see at a particular interval? And there were other questions that dealt more with, how much do you trust this person? More of a social connection with this person. And it turned out that this effect that we observed seemed to really be driven by the questions about contact intimacy. So the relationship with cognitive function and the relationship with salience network functional connectivity was really much stronger with the contact intimacy questions. I also want to note that work by Nathan Spreng and colleagues asked a similar question, but rather than looking at social engagement or social isolation per se, they asked about loneliness. So we can think of loneliness as the subjective experience of social isolation, so how alone someone feels. And the Lubin scale is a bit more of an objective measure of social isolation. Of course, it's still based on participant self-report, so it is by nature subjective, but it does get a more objective concept of social engagement, more focused on how many people they engage with and how often, and less about how they feel about it. So this work by Spreng and colleagues found that default mode connectivity differed between those who reported feeling lonely compared to those who did not. And so taken with our work, it may be that salience network connectivity is more sensitive to these more objective measures of social engagement and social network size, number and frequency of social contacts, while default mode connectivity is more sensitive to subjective feelings of loneliness. That's not something that we tested directly. It's not something that I've seen explored, but I think it is a potentially interesting distinction and does make some intuitive sense. So what I would love for you to take away from this talk, first and foremost, is that brain connectivity is deeply relevant for normal and pathological cognitive aging. And incorporating measures of brain connectivity into your research may provide more complete answers to your research questions, and it can open new and interesting avenues of inquiry. The other point that I want to make is just highlighting the incredible value of the Michigan ADRC. It's really fantastic to have them here and to be able to work with them. Collecting, of course, I think about it from a neuroimaging perspective, but having this rich multimodal neuroimaging data on each participant, with neuropsych testing and biomarkers collected longitudinally, really takes an incredible amount of work, and really allows us to ask some interesting questions. And whatever your interests are around cognitive aging and dementia, there's something there that they're able to provide that can really deepen your avenue of research. So if you're interested in incorporating neuroimaging data into your research questions around cognitive aging and dementia, reach out to the Neuroimaging Corps, start a dialogue. as I just showed, I am an example of someone who did that, and we used it to answer a research question that we had that was then used in subsequent work that Hiroko and I did. So would highly recommend, if that's something you're interested in, go ahead and take that dive. Before I get to acknowledgements, I do also want to mention, there are lots of additional resources that are available if you're interested in learning more about brain connectivity. As Stephanie mentioned, next month someone will be presenting on diffusion weighted imaging. I provided a fairly superficial overview. I'm sure they'll go much deeper, and hopefully I didn't make any false statements that they have to correct. But if you're interested in learning more about structural connectivity, there are talks like those that MADRC puts on that are available to you. There are talks on their YouTube channel. I was watching one by I think it was Drew Bender showing some of his work with diffusion weighted imaging that was really fantastic. Again, what I showed here is more of the basics, and listening to him and the work that he does was very, very interesting. Would highly recommend it. So with that, I'll start by highlighting my current program, the research program on cognitive and neuromodulation-based interventions. Thanking Ben, Eileen, Alex, and Quentin for their support and in some cases ideas for this talk. So this program looks at brain stimulation as an intervention for cognitive decline. And so hopefully in the near future, you may see some talks from Ben or Alex or Quentin or myself that use brain connectivity data. And from having seen this talk, hopefully you'll be better prepared to receive and interpret those results. Also want to thank Jeske and Hiroko for their mentorship on the project that I showed the results for. Hank and Bruno and Ari at MADRC, again, for supporting that project and making it possible. Again, providing the funding and the data, really, really supported it from start to finish, as well as Scott. I want to thank the summer series team for putting this on and the opportunity to speak here and helping to make things run so smoothly, as well as Elise Auerbach Pruitt, who provided feedback on the talk. It's actually our anniversary today, which is perfect timing because, as you all know, the 7th anniversary is traditionally the webinar anniversary where couples exchange webinars. So this is mine. Mine was all ready to go. Timing was perfect. And with that, I am happy to take questions during the remaining time.
Okay, I do have a question in the chat real quick. It says, thank you, Dr. Pruitt, great talk. I have 2 questions. One, will we have the slides somewhere? Yes, Violet, if you email me, Stephanie Nava, I have the slides. I can give them to you, or I can just give them to you right after this. Let's see. Have you tried to include structural data into the analysis?
Yeah, that's a really great question. And I have not. So, it would be a good thing to do. I haven't done it. Structural and functional connectivity can be, somewhat associated where, greater structural connectivity can help to facilitate greater functional connectivity. That's not always strictly the case, but the inclusion of structural connectivity metrics into the analysis that we looked at regarding social isolation would be interesting. It's not necessarily my area of expertise, so we didn't prioritize looking at that. Uh, but it would be interesting both to see if it's related to social isolation, as well as putting into the model in the context of the functional connectivity. So the very short answer is no, I didn't. But yes, it would certainly be an interesting question.
All right, I have a question in the Q&A. In your study, did you statistically adjust for the potential confounding effects of COVID-19 on the social engagement data? Furthermore, do you believe that COVID-19 could have long-term impacts on aging-related neuroimaging data that might influence your research or other connectivity type research in the future?
I love that question. That's a great question. We actually, the data that we worked on and that was presented here was all collected prior to COVID-19. So the particular results that I showed here would not have been impacted by COVID. The subsequent clinical trial that Hiroko was running and that I did some analysis for, COVID hit right as we were starting to collect some of the follow-up data in that clinical trial. So the neuroimaging sample size in that clinical trial is unfortunately very small. And so some of these questions that we were interested in terms of social isolation as a modifiable risk factor for cognitive decline and how functional connectivity ties into that. When COVID hit, obviously scanning stopped, and so we were not able to expand on the work that we did here in the clinical trial. We did analyze the smaller sample that we had and did find some interesting results. But I think to your point, the fact that COVID occurred absolutely did have a substantial impact on social isolation in our population more generally. And I think particularly in older adults who are at high vulnerability for it anyways. Many words have been written about this. But I would just say that it's interesting to think of in the context of, sort of a cohort effect, that there is a cohort of older adults who are going to be differentially impacted by, the pandemic in terms of their social isolation, that older adults before them and older adults after them, hopefully don't want to jinx us another pandemic, but the people who are older adults during that period, experience this additional social isolation in a way that other older adults haven't and will not. And so it's interesting to think about, collecting data from these current older adults through that lens.
Right. Thank you very much for the great talk today. It was very informative, and it was nice to follow around along with for somebody who doesn't have such a background in imaging. So thank you very much for that great talk. I did put the evaluation in the chat for those of you who stuck around. Please go ahead and take the time and fill that out. And thank you so much for coming out today. And I hope you join us on August 7th for our next neuroimaging summer series. So thank you so much.